The virus first attached to cells in John's body that contain a molecule known as CD4 and released its genetic material into the host cells. They include macrophages, dendritic cells, and T helper cells. Through viral replication and transcription, the genetic material incorporates itself into John's genome. Inside the cell, the viral enzyme, reverse transcriptase, converts a single-stranded RNA from the virus into double-stranded DNA that is incorporated into the genome of the host cell.
When John's cells became invaded by the virus, his body's natural immune response was activated to protect itself from harm. One of the key role players of immune response is inflammation, which is usually responsible for walling off areas of injury and facilitating the healing process. Under normal circumstances, the immune response is self-limiting, but inflammation can become chronic if the trigger persists or if suppressive control mechanisms do not work properly. In the case of HIV, the body is unable to completely eradicate the infection and thus inflammation is sustained. Along with this, John's infection has resulted in a secondary condition known as hyperlipidemia, which refers to increased levels of fat in the blood, shown by his test results. In the case of HIV, this tends to be present as a molecule low-density lipoprotein, a class of cholesterol closely linked to atherosclerosis and cardiovascular disease. The mechanism for the development of hyperlipidemia in HIV-positive patients is unknown. However, research suggests it could lie in the HIV virus affecting fat distribution in patients, or alternatively in the action of certain antiretrovirals. John's constant state of low-grade systemic inflammation, coupled with the high fat levels in his blood and the action of the HIV itself, has led to damage of his blood vessel walls. This makes it easier for LDL to enter the tunica intima of his blood vessels and cause the formation of plaques. Recruitment of inflammatory markers to the site of damage further worsens John's inflammatory state and can cause plaques to become larger and possibly more unstable, narrowing his vessels more and more and increasing stress on his circulatory system. John's HIV status and his steadily decreasing CD4 count has also caused his body to respond by producing more of the other cells responsible for immune response. Among these are monocytes, cells which have been linked to atherogenesis. Again, the mechanism for this is still under study, but scientists hypothesize that it could be due to increased monocyte activity aggravating the plaques, or that with HIV infection, monocytes differentiate into a subclass that intensify inflammation and plaque formation.